Welcome to my aspect in BCH 314. I'll be taking the general properties of amino acids, and we all know that amino acids are the building block of protein. All amino acids are very essential in order for you to, in order for are very essential or important for the protein formation. So your bigger proteins such as insulin are combinations of several amino acids in order to form them. So it is very good for you to understand what amino acid, how you can identify an amino acid, and what are the reactions that an amino acid can 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 be involved in. By identifying those reactions and properties of amino acid, you can be able to tell what kind of compound best present. Is just an advanced application of test of metal ions that you do in what's it called in basic chemistries. And in order for you to test for amino acid, there are also different tests that you can reaction or that your amino acid actually carry out. But for you to be able to um, go about this, you must understand the basic chemistry about amino acid. What are the key characteristics or components of an amino acid? Now, in a basic amino acid, the simplest amino acid, which is glycine, we have two hydrogen, we have an amino group and a carboxyl group. Now, in the image above, you see that there is an R group. Now, for every other form of amino acid, the R is substituted with different groups or different compounds that will form the amino acid. So, for an amino acid, it must have a common functional group. That is, it must have one alpha amino group and one alpha carboxy group. These are the basic or the simplest amino acid. And like I told you, I gave you an assignment to look into what's it called, the different classifications of amino acid and master their structure, because you tend to understand or how to use them in the future. And also, it's also good for you to know the the symbols used for this amino acid, so that when these symbols are being given, you can be able to project that this is the particular amino acid that is being talked about. Then another, okay, now for the one of the reactions that amino acid take place, amino acid leads to salt formation. So it, like every acid, when acid reacts with base, it leads to what? Salt formation. So amino acid also is an organic acid and also react with hydroxide, metal hydroxide or hydrazide to form salt formation with the releasing of water. Also, it also reacts with alcohol to yield esters. That is, it is also called esterification. So this is amino acid reacting with an ester under an acid catalyst, and we have ethyl ester of an amino acid with the release of water. We also have reactions with amines to form amide compound. That's the reaction. Decarboxylation. So amino acid, remember when I told you the basic what's it called? The basic structure of an amino acid is it has an amino group and it has one carboxyl group. So this amino acid can also undergo decarboxylation, like histidine undergoing decarboxylation to form histamine, and with the release of CO2, tyrosine to form tyramine plus CO2 and the likes. We also have reaction with mineral acid. Amino acid can react with ACL. It can also react with benzanamide. So you can see the reaction. It leads to the formation of Schiff base, which is an intermediate step for transaminase and transamination reaction, which you have in your urea formation, in the urea formation in the pathway, in its pathway. Also have Sangas reaction. Sangas reaction. This Sangas reaction is very important in detection of amino acid in a particular situation. You can use it to quantify amino acid present in a particular situation solution. For example, if your urine is being collected, you can carry out Sanger's reaction in order for you to determine the numbers of amino acid that have escaped from your body system that are present in the urine. These Sanger's there is a Sanger's reagent, and when it reacts with an amino acid, it gives a yellow color coloration, and this can be measured through your spectrophotometer. And I think that so. And what happens in this reaction is that the sugar, the reagent frees the amino group in the polypeptide chain. It's just like a scissors going about at the amino group joining with the carboxyl and cuts it. Then with that you can use to calculate the number of amino acid present in a particular solution. We also have formation of peptide bond, and remember your amino acid has a carboxyl group and an amino group. So the formation of peptide bond is between the carboxyl group of an amino acid with the with the amino group of another amino acid, thereby forming uh, di forming dipeptide bond. This is a covalent bond with the release of water. Now, when you have a peptide bond, two peptide bond between three amino acids, that's called tripeptide bond. Tripeptide bond. We also have neoadrene reaction. This is an oxidative decarboxylation reaction. This is also sensitive for the detection of amino acid, just like the Sanger's reaction. So please identify these key reactions for determining the amino acid concentration. We have Sanger's reaction, neoadrene's reaction. So you should be able to understand the reaction and how what product is being formed. We also have Zutation. This is a situation whereby the charges on an amino acid neutralization order. We have the amino group providing with an overall charge of negative. That is the amino group neutralizing the carboxyl group, so that the amino acid has a neutral charge. Then let's look at the chirality of amino acid. I know you are very conversant with this chirality of amino acid. Before I go further, know that we have the basic amino acid that are found in proteins are 20. And those are the 20 I asked you to master. In nature, we have about 300 amino acids. These are amino acids that are present in nature. They are also servers. Some of them are therapeutic in nature. They are found in antibiotics and the like. So now we are going to look at chirality of amino acid, and what these all these 20 amino acid are all of them are chiral in nature. The answer is basic no. Why? Because a chiral carbon must have 4 substitutes or 4 different groups around it. So therefore, glycine does not have 4 different groups around its center carbon. So glycine is not a chiral carbon, while the 19 other amino acids are chiral carbon. So look at this chiral object. This first set of image are super impossible by rotation. Super impossible by rotation on the mirror. But when you look at the other one with the big brown mug, you can see that the image in the front of mirror is able to read it. But these are what we call chiral objects, and a chiral object, I've given you the the definition of chiral object. And another thing that you need to understand is what they mean by stereoisomers. These are molecules of same chemical structure with different which differs from each other by their 3D conformations. They are just called optical isomer because the image have changed them. That's why they are called stereoisomer. in the presence of image, which we will also discover down the line. Then we also have enantiomer, which is pairs of stereoisomers which can be superimposed by mirror uh, uh, mirror mm, mirror reflection. Now, looking at stereocarbon, and depending on the chains or carbon present in an amino acid. An amino acid can have more than more than two two chiral carbons, and if you want to calculate the total the total chirality of an amino acid, look at the chiral carbons. We have four different groups around it, then 2 raised to power n will give you the exact numbers of stereoisomers that are very possible in the what's it called in the image. So okay, I've talked about glycine. For the case of glycine, we cannot have L, we cannot have D isomer. Well, the L is level rotatory, and the D is destro rotatory, and so these are very important for you to understand. And when you look at the L, just look at L as life. These are found in living organisms, while the D are commonly found in nature. Most especially, they are found in bacteria. They are found in bacteria. They are found. They are not really found in nature, but in some cases we have them in the what's it called in the bacterial cell wall. We have them in antibiotics. We have them in neurotransmitter in invertebrate mammals. So invertebrate organism. So these things are very important. Why am I talking about this stereochemistry? The significance is that, number one, the specificity of an enzyme is very important. So for an enzyme to be stereospecific, stereospecific, it has to, I said level rotatory L form of amino acid is what's it called occurs in living things. So for an enzyme to act on any protein, the protein must have L amino acid for it to be stereospecific in nature. And for protein folding also, it has to be what's it called it has to be held in nature. Then it's also good for protein drug design. There are some what's it called there are some amino acids that are very what's it called that are very inactive in their level rotatory, and while they are therapeutic in their destro. So it depends on them. Like I said, they are found in antibiotics, and they are being modified in order to be able to carry out the specific functions of functions that one desire it to have. A knowledge of this chirality is very crucial for modifying enzymes or creating synthetic peptides in the organism. By the time there is what we call modifications of protein, most especially when you want to use it for application products, or you want to use it for treatment of particular diseases or genetic diseases. One has to look at the destro or the stereoisomers of this compound. And with this, we come to the end of my aspect. If there are any questions, you can type it below. I will come and answer them once I'm charged. So I will also send this material to you on before I come to your class.